Awesome, thank you, Mike. And yeah, I'm very excited to give everybody an update on modified bases and methylation. So let's get right into it. Awesome. So I'm going to be giving you an intro to modified bases and methylation in Nanopore, touch on the production models that we have, what's coming next, and modified base tools for downstream analysis, which is a big new focus for us now that we've sort of grown the catalog of modified bases. All right, so modified bases in Nanopore comes for free with the technology, right? The signal has the modified bases in it. We just have to be able to pull them out. So there's no extra prep required, nothing, no extra runs. It's one run, your modified bases are in there, and we can now accurately detect natural modified bases, 5MC, 5HMC, and 6MA. Along with that, you get all the other advantages of Nanopore. So because there's no other prep, that the reads aren't shorter, it's in the native data that you already have. You just need to pull it out. Yeah, everything that you get, real-time sequencing, everything else is there. Also on the right here, we'll use a lot of plots that look like this. So the blue is going to be the canonical bases, red is 5MC, and the pink is going to be the 5HMC. So these are sort of IGV screenshots showing the reference bases along the axis and reads along the y-axis there. Awesome, so how do we call these modified bases? So we have Remora is the style of models that we've chosen, which is modified bases on top of base calling. So that means that we run the base caller through, and then you predict the modified bases on top. So at the top here, I'm showing you a squiggle, the raw signal traces, and what you're looking for are these signal deflections between the canonical reads in black and modified bases in red there. So the Remora fish latches onto larger aquatic creatures and performs different roles, data and protection, with very little drag and feeds off the host inputs. This is sort of an analogy onto the Remora algorithms, which latch onto the base callers and perform modified bases separated from the algorithm, which has these advantages of higher accuracy and quicker to train. So that's the Remora algorithm. So to give you a little bit of detail, I won't go into all the details of how these models work, but you bring in the full read with normalized signal, and then you chop out the bits that you want to call, and you look at those bits of signal, the base calls, and where the base caller put those bases within the signal, and then you digest it into a form that the neural net can take. And this is, at a high level, how the Remora models work. I've given a bit more detail in previous talks, so I think last NCM I gave a little bit more detail on this to jump into there. I want to jump in real quick and highlight two of the main data types that we use. So on your left here is the randomer data. This is how we train most of our models. And there's a beta, which is beta the fish. Everything has to be named after a fish. So you can express your interest in this repository with the QR code here. What this is we print these molecules that have random bases and then surrounded by fixed bases with a fixed position of modified bases or canonical bases in the middle. So you have unknown sequence but known modified bases, and we use the duplex technology to get our reference bases in this section. You can also work with simplex. I'll get into that as well. So this is the main key that's driven our ability to give you all context models. And has been a really powerful data source for us that we've continued to sort of expand how we do modified bases. On the right here, I'm showing the ground truth oligonucleotides. I'm going to be showing a lot of accuracy metrics. Our accuracy metrics are per read, per site calls that we've printed validation strands. So there's no fancy statistics or anything. You just take what you see in the bottom there. You count up how many modified bases you see and how many canonical calls you have, and you sum that up, and that's our accuracy. It's per read, per site. There's no aggregation. That's the accuracy of the calls that you get in your reads. So I just wanted to highlight that one there. All right. So moving on to our production models. Our production models, we've moved from context specific to trying to do as much all context modified bases as we can, so that you get your you get your modified base calls at every A or C or any other base that we produce as it comes out. So again, we're looking at per read, per site accuracies in this confusion matrix here for our highlight model, the all context 5MC, 5HMC model. And then the accuracy gains in the base caller have actually improved the modified base calls as well. So getting more modified base samples into base caller training has improved the base caller accuracy around modified bases, and that's translated into better modified base calls as well. Additionally, we've also included modified bases near each other so that you know what a canonical and modified bases look like near another modified base. So we've been able to include those in training as well. Additionally, we have CPG context models. That's sort of the bread and butter that we started with. And what we're doing now is we have the all context models, and you can grab the CG context out of those. It runs a bit more compute efficiently. and we get much higher accuracy. The CG context gives a little more signal diversity, and it makes the calls a bit easier. And then additionally, using our ModKit tool, which I'll get into in a minute, in the bottom right, you can take this 5MC plus 5HMC calls and condense them down into 5MC calls to give you a comparison to classic bisulfite or other technologies. So that you can still condense your data down into that, and you see the accuracy is well above 99% when you condense it down there. Finally, the next all-context 6MA is the last production model we have here. And again, we're targeting really high accuracies on these. And again, you can see the picture of the control strands here with the green signifying the 6MA calls, and the control strands are really high accuracy canonical calls as well. All right, so research model that we just released today, about an hour and a half ago, is a 4MC and 5MC for the bacterial crowd. So this has been one that we've gotten a lot of requests to get a 4MC model. So you can see here on the right, on the left here, we have our per read per site accuracies. We're aiming to improve the per read per site accuracy of the 4MC, but we still believe that this is a very useful model for the community, and that there's a lot we can do with it. And on the right here, you can see the known modified base motifs for 4MC, 5MC, and 6MA. All of this run with one Dorado call, and you get all of your modified bases. I had to change the colors a bit. The canonical is no longer bright blue because it made it hard to see the mods. So we really appreciate all the work from the community where to visualize these modified bases. All right, so what's next? So we have DNA damage. So ADOX-OG shows up in oxidative stress, and ribobases in DNA, which has shown genome instability, and we've done proof of concepts, and we're working towards models for these. So we've shown that you get signal deflections in these, and we're working towards getting models created for these. In order to create these models, we're building out the Remora toolkit to handle some of these tougher, larger signal deflections that can cause certain issues in training, as well as we have base pairing here with ADOX-OG that incorporates the different canonical base on the opposite strand, so there's fundamentally a different base at that central position in a lot of the reads that we print. So working on building out the toolkit to handle and train these bases. Finally, a big step that we've made is working on RNA modified bases. So Ino's talk next will go into a bit more detail here, but we've trained these RNA models from simplex randomers, and we're getting really high accuracy. And now that we've released a model for M6A, we're expanding that as well to pseudouridine, 5MC, and inosane are our sort of next targets on RNA bases. All right, so now that we've started expanding the catalog of modified bases to a number of the ones that people want. One thing that we're really trying to focus on in the mod base group is the downstream analysis. So I'll go a bit into ModKit and Remora and how these things fit together and how we envision helping everybody with the downstream analysis, which we know has been a bit of a pain point with doing modified bases. So Remora is used to train modified base models and do some signal analysis. So that's sort of the core high research project for everybody that produces the modified base models, which then get ported into Dorado for production base calling with modified bases. ModKit is the post-processing after you've called modified bases. ModKit is the tool for anything downstream, and I'll go into all the things that it can do in a minute. You can use it in a pipeline, so Epitome uses this, as well as any summary statistics, high-level things you want to get out of your mod BAMs. And you can also use it for exploratory data analysis to get read-level modification calls, differential methylation, and anything else downstream. If there's more things people want, please let us know. So one of the main things that people want is to see a pileup. When you have a mod BAM, you want to see, is there 5MC, HMC at my reference sites? So this is, and it employs high accuracy filters. So we filter out about 10% of the reads, showing the lowest confidence modified base calls in order to give you the highest accuracy results in your pileups. So this is the most common first step that people want when they have modified bases. So that's the core algorithm that we have here. The next one is differential methylation that was added recently. So in here, I'm showing two examples here looking for haplotype differences in HG002. So you can see you have one haplotype canonical and the other completely modified. And you can do this on single sites or entire regions shown on the right here. I would like to highlight Yuliel Fu, who is a collaborator with the applications team internally. He'll be giving a talk tomorrow in the bioinformatics breakout session on methphaser. So you can sort of pick out in this last unphased bin of reads on the right here, the reads that are modified that belong in haplotype 1 and haplotype 2 just for methylation. And we're looking at this more in our research group as well. But a very exciting breakthrough to really give everybody the power of modified bases. One final feature that we're really excited about is hemimethylation. So when you do a duplex read, you read over one strand and the reverse strand. Those are combined to give you the canonical sequence. But you also get modified bases from the forward and reverse strand, and this can be output directly by the base caller, and Dorado 0.5 now has the ability to output both of these at the same time, and then ModKit gives you the ability to pile these up in the same idea of piling up, and you can see where you have hemimethylation in the core molecules. This is really exciting. One of the things I've been looking forward to for modified bases for a long time. Finally, I'm skip over this pretty quickly, I'm out of time, but we have a Remora 3.0 update where we have done a dataset rework. You can train efficiently from 1 billion training examples, flexible config-driven datasets, which is how we've done all of these big updates. Full RNA support and the signal visualizations you see on the right, as well as an improved metrics API giving you the p-values on the bottom here. Awesome. And yeah, so this is everything I talked about. The production models, our new research 4MC model, and a new frontier for RNA modified bases, continuing to expand the catalog of DNA, ModKit enabling our downstream analysis, oh, that was an old version. That will be talked about tomorrow. So look forward to that. Thank you for everybody else for listening.